I'll start with an overview on genetics in ophthalmology. I want to say that I have no financial or other interest in any of the manufacturers of genes. At first, the literature for scientific genetics in ophthalmology was in the research journals, IOVS and Ophthalmic Genetics. But as more and more interest evolved, it hit the mainline journals in ophthalmology, iNet, which is a publication of the American Academy of Ophthalmology. Archives put out 2 issues, front cover, and ophthalmology of the American Academy. Even neurology got into it. And then it hit the popular press with Newsweek magazine and Time magazine, each showing it on their front covers. So why do we have to know about genetics? Because our patients know about genetics. And they're going to ask the questions, and we've got to know the answers. To show their interest in genetics, some patients actually, for artwork, have patterns of their genetic DNA electrophoresis on their walls. And this person put his dog's beagle DNA electrophoresis as artwork on the walls. Moreover, some owners of dogs that love their dogs, and when a dog dies, they'll take a piece of their skin, DNA, and implant it into the cell of another dog, put it in still another dog for maturation, and he now has 2 dogs of his loved original dog. Parents are checking the DNA of their offspring to see if they have the acne 3 variant R, because this gives very sporadic sprinting type of muscle tone, making them runners. That variant of the acne 3 gene, the R variant, makes them sprint runners. If the X variant, the protein is different, and it's not good for that. With that, did Santhi Soundarajan, who won the Asian Games in 2006, was she a he, or did she have the R variant? She wouldn't let them pull down her pants, so she lost the gold medal. Well, Caster Semenya, the winner of the 2009 World Games, also suspected of being a male, did allow them to pull down her pants, and she kept the gold. But really, must a runner have to pull down their pants to prove their sex? Notice the 46 for the chromosome genes. When all they have to do is have their DNA checked with an oral mucosal swab. Now, everyone is interested in sex, so I'll just start my talk with this. The SRY gene, which is really responsible for sex determination, is on the Y chromosome, not at the top, but a little further down. Here's a little magnification of it. This is where its location is. And that has significance because in meiosis you get a frequent exchange, and if it's just the tips that's exchanged, it doesn't mean anything because the SRY gene remains on the Y chromosome. And if the whole thing is switched over to the X gene, well, if it stays in the same person, it doesn't mean anything. But in meiosis, it could go over and be more confusing. For instance, here we have the SRY gene on an X chromosome, and so you have a female with male characteristics, and here we have a male without the SRY gene. So you have a male with female characteristics. But let's take an individual who's otherwise genetically okay. And I need help here. Help! You take someone with a genetically proper setup, but still they may have the characteristics of the other sex. And that's because of the adrenal gland. If there's insufficiency, lack of testosterone, you can get an effeminate a male. If you have an overaction of the testosterone, you can get a masculine female. So it gets confusing. I want to go into this now because it shows the fascination that people have with genetics. And when the episodes of King Tut came out a few years ago, National Geographic had on the front page the DNA of King Tut. And it was very difficult to extract that DNA. I'm not going to go into it too much. But what they put together in the neighboring tombs, the neighboring mummies, they were able to identify the genetics of the family. They got the grandparents, the parents. They have a woman who they think is the mother. They just call her KV3 from the cave. And the father they knew. With that, King Tut was born. But there was a half-sister. Now, who was King Tut's wife? Well, through this genetic analysis, remember that this was old DNA, they found out that King Tut's wife was actually his half-sister. And with that, they produced a stillborn girl. More recently, the country became fascinated with genetics with the assassination of bin Laden, because was the person that was assassinated really bin Laden? Well, the New York Times wrote this up, and they just didn't say that a DNA analysis was done by collecting the DNA samples from known relatives. No, they went into a whole description of DNA. This is what our sophisticated patients are reading. They talk about DNA sequences, same as everyone else. The test is needed, focuses on a small number of locations in the genome, 13 to 16 spots, etc. It talks about junk DNA, and this junk DNA does not build muscle or bone, but it's used for analysis. It goes further. And it talks about strings of nucleotides called short tandem repeats. Who in this room knows what a short tandem repeat is? Stay tuned. After this message, I'll tell you. Now, the failure of the human genome to really predict what a person's future illnesses would be, or what your appearance is, or what your overall makeup is, was highlighted in this article from the New York Times a few years ago. Now, the human genome sequence was actually sequences of the known genes at that time. And this study hoped to pinpoint the DNA changes responsible for the common diseases. But it was disappointing, because the genetic variations really don't explain the links of the genetics to most diseases. Why? Because it could be polygenic, with a lot of diseases, cancer and diabetes, by various genetic and epigenetic variations, and multifactorial, environment, hormones, etc. So it's not necessarily just the genome. It counts, but it's not exclusive. And then you have in the sequence, this is the insulin gene, the gene for insulin, the silencers, enhancers, promoters, very complicated issues for how a person will eventually evolve, and the disease states that could be inherited. To make things more complicated, here's LCA, Leber's congenital amaurosis. I'm just using this as an example, where it could be caused by many different genes, and many of these genes can cause different dystrophies. Here we have combination to retinitis pigmentosa, and several of these genes could go together for cone rod dystrophy, and different genes also for retinitis pigmentosa. So you get whole combinations and sequences. So what can genetic testing really do? It could predict a medical condition, diagnose some genetic disorders, most important, beneficial drugs for a particular person, and maybe the inheritance of genetic diseases. So the purposes of genetic studies is to get to a point where a patient's genetics could allow for development of specific therapeutic strategies for that person. And there's a whole batch of different types of gene therapies, antisense, recombinant drugs, which is currently being used for insulin, human growth factors, etc. And this will be discussed by our panel. Gene therapy, one type, is vectors, the adenoviral associated viral vectors. These viruses have been altered genetically to remove the bad DNA from a person's cell and replace it with good DNA. And to show the interest in this current issue of Archives, which I alluded to in our previous course for another reason, actually talks about gene therapy for Leber congenital amaurosis. There were a series of patients, 15 children and adults followed, where they injected subretinally the gene to combat LCA. And this will be alluded to a little bit later by Dr. Demiraj. So how does this antisense work? Well, they're small nucleotides, antisense, which I'll go into in a minute, that clamps on to a person's messenger RNA and interferes with the development of a disease order. So let's go into Genetics 101, the crash course. All this is in your handouts, but I want to show you that only 1 to 5% of base pairs code for protein, and the rest are the junk DNA. There's about 35,000 genes in a haploid, a chromatid, and the difference in the genome from one person to the other is only about 1/10%, and we're only 2% different from a chimpanzee, our best friends. This is in your handouts. I want to highlight this because a centimorgan is one of the methods of calculations, and that means that there's in meiosis a crossover about every 100 times. And it's about 1 million base pairs in length. And this is important for recombination in meiosis. Here's the simple meiosis, where the offspring can get chromatid for maternal and paternal, but moreover, the crossover. So you have a mixture of maternal and paternal plus the crossovers. And here's a very interesting one, chromosome translocations, where instead of being homologous chromosomes crossing over, different chromosomes can cross over. Here is chromosome 4 crossing over with chromosome 20. You can see how it gets confusing for any particular individual. Now, here's in the metaphase, the center is the centromere. Through Giemsa staining, you count from the centromere out. The long arm is the Q, small is the P, telomere at both ends, so we count from 0 centromere down and up. And of course the spread, where the larger chromosomes are at the beginning, smaller at the end, except that 21 is a little bit smaller than 22, and then you have the sex chromosomes. Now all of these chromosomes, they're wrapped up, the DNA is wrapped up in a helical fashion around histones, which are globules of proteins. And all of these histones are wrapped around and form the chromosome in a helical fashion. This first came out in 1953. Watson and Crick that first described the double spiral helical formation of DNA. Before it was considered by Planck a single strand. This was in Nature, 1953. A month later, in May of 1953, they went further and talked about the replication of the chromosomes in DNA. So you could always go back to this 1953 Nature magazine. It's very clear. I'm going over now into a different type of DNA, that of the mitochondria, which are actually DNA smaller, derived from old bacteria DNA. That was the origin, and a lot of the DNA of the mitochondria in evolution eventually became engulfed in the nucleus. And we know about mitochondria, energy, metabolism, and regulates apoptosis. But I'm going to go further in that. There's only about 2 to 10 genes per mitochondria. Maternal inheritance, that is the key that we have to know. And because it's used in the identification of human remains, which we're going to go into. Of the 37 genes, look at it this way, 13 protein genes and 2 RNA genes, but in ophthalmology, we have 3 of them. Leber's hereditary optic neuropathy, don't confuse this with Leber's congenital amaurosis. Leber was very busy with this, lots of things named after him. Chronic progressive external ophthalmoplegia and Kearns-Sayre disease, all maternal inheritance compared to nuclear, which could be from both parents. And here, why is it maternal? Well, the mitochondria are in the tail of the sperm, which usually break off. It cannot penetrate the egg. So the mitochondria that's left are really maternal and not paternal. And here's inheritance, where we could see maternal, only nothing from the pop. All right, I said that you'll see how it is in inheritance. Another article, this is big. Any Australians here? Because Australians know about Ned Kelly. Ned Kelly was a legend in the end of the 1800s. He was like Jesse James. He was the one who fought against the British, and he did lots of things. And he also was a criminal, a fugitive. So when he was finally caught, he was hung, and he was buried. But his remains disappeared. And now they were looking to see, when they found a skull, if indeed that was his. So here's the study that was done. And this is published. Your patients are reading this, at least in the New York Times. They found Lee Ulver, who was an art teacher. And it was known that he was descended from Ned Kelly's mother through a direct line of women. So they tested his mitochondrial DNA. And lo and behold, for that skull that they found, it wasn't Ned Kelly's. But they found these bones that were his, from the maternal DNA, and they found a piece of his skull. But now, whose skull did they find? Well, a few years later, another fugitive was found, Frederick Deming. Anyone know who Frederick Deming was? He was Jack the Ripper. And they now are looking for maternal inheritance for Jack the Ripper to see if that's his. So you can see the interest that the people have in genetics. Now, all of our life are based on 4 bases: the purines, adenine, guanine, thymine, cytosine, the pyrimidines. It's all color-coded, which I'll tell you. Now, this is carbon-based. Carbon-based. But does it have to be? Really? Do we have to go to some distant galaxy or some planet or moon to find some primordial pond that has maybe a different basis of, let's say, sulfur or iron? That's the basis of life there, in order to find some extraterrestrial figure with a different makeup? No. All we have to do is go up the street from where I live in San Diego to the Scripps Research Institute, where Gerald Joyce is working on new life forms, because the definition of life is a sustaining system capable of undergoing Darwinian evolution that could replicate and evolve through mutations. And he's working on this, and this is just 2 bases. In Florida, they're working on other systems that there are more than 4 bases. Oh no, we're not done yet. I have 20 minutes. And I like the noise, though. It keeps me awake, because God forbid I should fall asleep while I'm lecturing. I don't want to go into that. And also, do we have to have just 20 amino acids? Do we have to have only 64 codons that code for 20 amino acids? Probably not. But this will be maybe the future. In the meantime, let's get back to the way it is now. In color coding, adenine is green, cytosine is blue, etc., and you'll see why. Now, the makeup of our nucleotide, the base is on carbon 1, and the hydrogen hydroxide is carbon 2, and the phosphorus bone, making it a nucleotide instead of a nucleoside, is attached to there. Now, I want to show you carbon 5 in cytosine, which you'll see in a second, because this has a lot to do with epigenetics, carbon 5. When we read our codons, they're 3-letter codons. We always start at the second letter. Forget the first one, the, owl, and cat, etc. It has to be in that sequence, because if you eliminate a letter, what's left, you still have to read it as threes. It makes no sense. And this genetic code will code for all of these amino acids. It's in your handouts. I want to highlight methionine because every protein has to start with this coding. It's only one. And remember that uracil is a substitute for thymine when you talk about RNA. It's to start coding. And the end, if you have a problem, an insertion, a deletion, etc., you get a stop codon, which is uracil adenine adenine, or uracil guanine adenine, or uracil adenine guanine, etc. These are the stops, and so you don't get a protein. Again, 3-letter forms. Here we can see how it codes for different proteins, or in this case amino acids, but you can get a frame shift. That's what it's called if you insert or delete any of these, and if you just do that, it makes no sense, and you get a stop codon, thymine, adenine, and you don't get the protein. Here, if you have a missense where just one of those bases is changed, yes, you have the same sequence, but you would get a different protein, and this is sickle cell disease, hemolytic anemia. That's called a missense, where you just change a base. That's different from a deletion, where one is deleted and nothing could be read because it doesn't make any sense, and at the end it's called nonsense because you're not getting anything, you're not getting a reasonable protein, and here's some of the diseases associated with that. In transcription, we have the introns and exons. The introns are eliminated when you get messenger RNA. The exons are spliced together, and that's called translation. And short tandem repeats, I'm glad you stayed here because now you get the message. It's a repetition of 2 to 5 nucleotides occurring in the intron region. It's eliminated, but it could be used for identifications, for genetic profiling, etc. And I have to get rid of that. I did. And once again, we have the sense, which is the original strand. The antisense is made from that. And then it's transcribed by eliminating the introns into messenger RNAs and translated into the transfer RNAs. And here is where it takes place in the nucleus, and then it spits out into the cytoplasm, and then you have the amino acids and proteins made. In pedigrees, the recent one is 4, and then you go back to the parents, grandparents, great-grandparents, and this is a case that Tony and I had together. I'm just using that as an example, where we had a certain CNA1, which is maternal. Look at this. This woman had 2 different partners, and still the children came out with the genetic abnormality. Showing it really came from her, but we couldn't go back any further, so she might have been the start of cornea plana type 1. I don't want to go into that, but the way it was done through Dr. Oldavi's lab, the DNA was extracted, heated, amplimeres added, cooled, heated, got the electrophoresis, etc., put through purification, and then we got the chromatogram, and this is what we got for cornea plana, where here's the normal control case, and we can see that from the adenine, cytosine came in, and that caused the abnormality, where we can see only a little bit of adenine, and because cytosine came in. Now they're doing microarrays in many institutions, larger surveys, newer techniques, faster, and here's the HiSec 2000, could read 3 billion bases per run. And since we have far fewer, you can imagine now how cheap it is, and now we're getting data deluge, and you can see how cheap it is now. Before we could, this is what it cost. 4 years ago, this is how much a sequencing cost, close to $9 million. Now it's estimated in a couple of years to be $1,000. I want to finish with epigenetics. Epi meaning over and above. Epigenetics is the study of hereditary changes in a phenotype without changes to the DNA sequence. Why are twins somewhat different? Why could some develop diseases and not others? Another thing, why with the total potent cell, parts become skin, parts become bone, parts become muscle. Now this goes back before Darwin to Lamarck, and he felt that traits acquired are a factor in evolution. It could be, look at this, the giraffe trying to stretch the neck. He feels that the stretching of the neck to get to the top of the trees caused the giraffes to epigenetically, without changing the DNA, to get longer necks. And the way it's done is that the methylacetyl group is attached to the histone through the tail, and then it attacks, once it's opened up, it attacks carbon 5 of the cysteine, of the cytosine base. This is what is worked on. And this could be inherited. So it just takes one generation to pass on to the others if it's in the sex chromosome, or it could end with an individual if it only takes place in the non-XY chromosomes. So it's methylation of C5 of the cytosine base. And here we can see the mechanism. It's in your handouts, and we can see how it attacks the tails of the histones and then unwinds it, and it could attack the cytosine. A very interesting article, Genomics and the Eye, appeared earlier this year in the New England Journal of Medicine. You may want to pull it up. Here it hit the general medical journal that everybody is reading, and they highlighted Genomics on the Eyes. Here's my references. It's in your handouts, and I thank you for listening and not falling asleep. I'll now turn the podium over to Dr. Aldavi.